Human Genome Project. The Human Genome Project is an international scientific research project with the primary goal of determining the sequence of chemical base pairs which make up DNA, and of identifying and mapping approximately 20,000 to 25,000 genes of the human genome from both physical and functional standpoint. Genetic makeup of an organism lies in DNA sequences. That means two individuals differs, then their DNA sequence should be also differs at least some places. This leads to be finding out complete DNA sequence of human genome. As a result, a project of sequencing human genome was launched in the year 1990. Human Genome Project was called as Mega Project. Human genome is said to have 3 into 10 to the power of 9 base pairs, and the total estimated cost of this project would be $9 billion US. The enormous data necessitated to use of high-speed computational devices for data storage, retrieval, and analysis. Human Genome Project was closely associated with the rapid development of new area in biology called as bioinformatics. The objective is to find disease-causing genes and probability use of information to develop more specific treatments. It also may be possible to locate patterns in gene expression which could help physicians gain insight in the body emergent populations. Human Genome Project main objective is to understand the genetic makeup of human species. It originally aimed to map the nucleotides contained in the human haploid reference genome. The genomes of any given individual is unique. Mapping the human genome involves sequencing multiple variations of each gene. What are the goals of Human Genome Project? 1. To identify 20,000 to 25,000 genes in human DNA. 2. Determine the sequence of 3 billion chemical base pairs that make up human DNA. Third one, to store the information in databases. 4. To improve the tools for data analysis. Fifth one, to transfer released technologies to other sectors like industries. Then final one is to address the ethical, legal, and social issues that may arise from the project. Thank you. DNA fingerprinting. DNA fingerprinting is a test to identify and evaluate the genetic information called DNA in a particular human cell. It is called fingerprinting because it is very unlikely that any two people would have exactly the same DNA information. The test is used to determine whatever a family relationship exists between two people, to identify organisms causing a disease, and to solve crimes. The chemical structure of humans' DNA is same. The only difference between peoples is the order of base pairs. There are millions of base pairs in each person's DNA, and every person has a different sequence. It is these differences in sequence of DNA which make every individual unique in their phenotypic appearance. DNA fingerprinting is a very quick to compare the DNA sequences of any two individuals. DNA fingerprinting involves identifying differences in some specific regions in DNA sequences called as repetitive DNA, because in these sequences a small stretch of DNA is repeated many times. These repetitive DNA are repeated from bulk genomic DNA, has different peaks during density gradient centrifugation. The bulk DNA forms a major peak, and other small peaks are referred to as satellite DNA. Depending upon base composition, length of segment and number of repetitive units, the satellite DNA is classified into many categories such as microsatellites and mini-satellites. These sequences normally do not code for any proteins, but they form a large portion of human genome. These sequences shows high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissues, blood, hair follicles, skin, bone, saliva and sperms etc. from an individual shows the same degree of polymorphism, they become useful identification tool in forensic applications. Thank you.